Welcome to Podcasts on Demand, a continuing medical education activity. This activity includes the most recent and current clinical data presented by leading experts. If you are seeking continuing education credit, please review the disclosures and the requirements for successful completion of the activity prior to listening to the podcast. A link is found in the podcast description that can direct you to this information.
Hello, I'm Dr. Eileen Scully from Johns Hopkins University School of Medicine, and welcome to our final clinical clip from CROI 2025. So I'm going to start off today by talking about one additional study on aging and inflammation. So this was abstract 182 on letermovir for CMV suppression. This presentation reported the more complete results of the available participants and had several salient observations. So first, letermovir did suppress mucosal CMV shedding through 12 weeks after interruption, and there was never any detectable plasma levels of CMV. Second, after that initial unexpected increase in soluble TNF receptor 2, over the remaining 48 weeks of the trial, they observed a decrease. They also observed a consistent decrease in IL-1 beta over the entire period of the trial, suggesting that at least some markers of inflammation did show a more consistent decline with treatment. The CMV or cytomegalovirus antibody levels declined during the trial and then had a slight uptrend after treatment discontinuation, supporting that there was a meaningful anti-CMV effect of the letermovir. And notably, the CD4 to 8 ratio substantially increased during the treatment period, with an effect that was more pronounced in the women in the trial and was associated with an approximately 70 cell per microliter increase in absolute CD4 count. So the clinical implications of this study are unclear, in particular because it was terminated early with such a small enrollment, but this analysis does suggest that the trial may actually have been stopped too soon and that there could have been a meaningful benefit from longer-term CMV suppression, and hopefully follow-up studies will give us some more information on those particular questions. So next, I wanted to highlight one study that was an HIV prevention effort specifically focused on people who have injection drug use. So in this study, investigators for HPTN 094 designed a randomized active control study evaluating the impact of mobile outreach units with pure navigation compared to the active control of pure navigation alone. And their goal was to improve care for opiate use disorder, HIV prevention, and among those living with HIV viral suppression. So the results were notable for amazing retention at more than 85%. And when they looked at only those individuals who had not died or were incarcerated, they had greater than 90% retention. They saw an unfortunately low rate of medication treatment for opiate use disorder at approximately 7% at their 26-week endpoint, and only about 40% HIV viral suppression. But they did see a decrease in the death in the intervention arm. So the estimated risk reduction for mortality was close to 70%. Although this was not a statistically significant result, the substantial magnitude of the effect points to the potential benefits of this approach and the need for integrated care for people with opiate use disorder. So now I'll move to a discussion of some of the late-breaking ART presentations in the special session. So first, there is data from the CARES trial. This is abstract 202, a late breaker. This phase 3 open-label non-inferiority study sought to evaluate the safety and utility of change from first-line ART to injectable long-acting ART, enrolling at 8 centers in Africa. So the initial results reporting non-inferior efficacy at 48 weeks have been reported previously, and here they report the extended follow-up to 96 weeks. At week 96, the primary endpoint, 96.9% in the long-acting arm had remained virally suppressed and 97.3% in the oral therapy arm, meeting their non-inferiority standard. 4 participants, or 1.6% of the long-acting arm participants, had confirmed virologic failure by week 96 and none in the oral therapy arm, and resistance was common at failure. 3 of those individuals resuppressed on dolutegravir 3-TDF, and 1 participant passed away from an unrelated surgical intervention. Overall, cabrelpivirine showed high efficacy, non-inferior to oral ART in a very public health approach with sparse viral load monitoring, and saw a low rate of confirmed virologic failure consistent with clinical trials and other real-world experience with this agent. So overall, very promising for the rollout of this approach in this setting. So now let's talk about some other novel ART approaches. First, there's another broadly neutralizing antibody study. This was abstract 203, also in the late-breaking session. The study evaluated the safety, efficacy, and tolerability of N6LS given every 4 months plus monthly cabotegravir for maintenance of HIV viral suppression. They enrolled adults with an HIV less than 50 copies per mil and phenotypic susceptibility to N6LS. Participants were randomized 2:2:1 to either N6LS intravenous formulation with cabotegravir or N6LS subcutaneous along with a hyaluronidase preparation that allows the accessibility of the agent plus cabotegravir, or continuing on their standard of care regimen. Confirmed virologic failure occurred in a total of 4 individuals, 2 in each of the broadly neutralizing antibody arms, and all of them resuppressed on initiation of oral treatment. Of the 4 failures with available data, in both of the intravenous group failures, there was resistance to the antibody at confirmed virologic failure. And in the subcutaneous group, one of the 2 individuals developed an integrase inhibitor resistance-associated mutation. So overall, the investigators saw better tolerance of the IV administration and will move forward with this formulation. Again, it's not totally clear how broadly neutralizing antibodies fit into the overall armamentarium. This particular broadly neutralizing antibody is anticipated to have more broad generalizable utility because of the high rate of susceptibility in the general population. But again, it is not 100% and needs to be tested in advance of using this therapeutic. So finally, I'll talk about 2 abstracts on switch strategies to doravirine and islatravir. This is abstract 204, and of which there are 2 parts. So in this particular trial, the investigators were evaluating the switch from bictegravir FTC-TAF to doravirine islatravir in this phase 3 study. Participants had virologic suppression for at least 3 months, no history of treatment failure, and no known resistance to doravirine, and were randomized to one of the 2 groups. And in this study, 2 participants on doravirine islatravir had a viral load of greater than 200. The first had a moderate viremia between 200 and 500 copies, despite tested expected levels of drug present in the plasma, and a subsequent genotype failed to amplify. The second, at week 48, had high-level viremia with no resistance-associated mutations and no drug detected. Both participants resuppressed when switched back to bictegravir FTC-TAF. Overall, doravirine islatravir met non-inferiority criteria when compared to bictegravir FTC-TAF. There was no treatment-emergent resistance, and no significant impact was observed on lymphocyte or CD4 counts. In the paired abstract, they looked at a broader strategy of switching from any baseline oral antiretroviral therapy, not just bictegravir FTC-TAF, to doravirine islatravir. This study enrolled adults with an HIV of less than 50 copies for at least 3 months on 2 or 3 drug therapy. Among the 500 participants, 40% were female and 60% were non-white. And the bottom line here was that doravirine islatravir was again non-inferior to baseline ART at week 48. 2 participants discontinued early after virologic failure at week 4 with multiple resistance-associated mutations, but they were both later identified to be ineligible for enrollment based on prior virologic failure or doravirine resistance at baseline. So 3 discontinued with virologic failure, 1 failed to amplify, 1 with resistance and undetectable drug levels, and overall there was no treatment-emergent drug resistance. So the safety profiles were generally comparable, although as is consistent with an open label switch study, there were higher rates of discontinuation on the doravirine islatravir option. And there was weight gain with the switch to doravirine islatravir. The investigators did do an analysis, and that weight gain was driven primarily by people who switched from a weight suppressive regimen, including either efavirenz or tenofovir disproxyl phosphate at baseline, and who were then switched to doravirine islatravir. There were no adverse events for lymphocyte or CD4 count, and no treatment-emergent resistance, as I mentioned, to doravirine or islatravir. So to summarize these 2 studies, doravirine islatravir is gaining data to support its use as a possible switch strategy, and has the novelty of not including an integrase inhibitor, and the similarity of other 2 drug strategies of requiring a really careful consideration of hepatitis B status. So thank you for joining us for episode 4. Please refer to the landing page to claim your continuing education credit, as well as to view our previous clips from the conference.
We hope you found this podcast useful and educational. To receive continuing education credit and to download your printable certificate, please go to the activity page at practice.cme.com to complete the post-test and evaluation to receive continuing education credit.